Speaker for the morning session is Dvir, from the Department of Molecular Microbiology and Biotechnology. That's a new name, isn't it?
No.
Will be. Okay.
Very soon will be changed.
And Tal will tell us about cardiac tissue regeneration from matrix design to bionic heart engineering.
Thank you very much, Ori and Adi, for inviting me to this symposium. It's a great honor for me. The talk is going to be less nanoscience, more nanotechnology. And the lab is working on building different types of tissues. We're working on spinal cords, brain, ovaries, bone marrow, but I will mainly focus today on building the heart for cardiac regeneration. So before starting to tell you about the technologies that we can offer, I'll say a few words about the disease that we're trying to cure. So first of all, heart diseases are still the #1 cause of death in the Western world. Actually, 1 out of 3 deaths in the United States is from heart disease. And myocardial infarction or heart attack captures a significant fracture of these diseases. And it happens when a major blood vessel that nourishes the left ventricle is blocked. As a consequence, blood, oxygen, nutrients are deprived from the ventricle. The cardiomyocytes, cardiac muscle cells, are very sensitive. They simply die when they don't get oxygen. And since these cells are special, they cannot proliferate, they cannot divide, and since we do not have enough stem cells in the heart, this whole area becomes fibrotic, becomes a scar tissue, cannot contract, cannot send pulses of blood to the rest of the body. And the poor statistic says that 50% of the people who've had a severe heart attack will die within 5 years. Currently, the only solution for the end-stage patients is a heart transplantation. And since we all know that there's a shortage in donors, there's a need to try to repopulate this area with cells that are capable of contraction. So the lab is working on different technologies, and I will mainly focus on tissue engineering. So what is the concept of this approach? We can take cells from the patient or from other sources. We cultivate them in a 2-dimensional flask, and then we need to create a tissue from these cells. And in the body, when we talk about tissues, it's not just cells, it's the cells and the extracellular matrix. And this matrix is the glue of the cells. So we see the cells in 3-dimensional biomaterials that we develop in the lab. These should mimic the natural matrix. We can add growth factors, small molecules, nanoparticles, and essentially we're doing everything we can to allow the cells to assemble into a functioning tissue that we can later on take and transplant back to the patient. So how do we create the cardiac patch? We can use different types of cells, either adult cells from biopsies or different types of stem cells. Currently, the lab is shifting towards working with induced pluripotent stem cells. These are cells that can be taken from the patient skin, for example, reprogrammed to become stem cells, and then differentiated to become cardiomyocytes, neuron, whatever we want to work with. Then we take these cells and seed them in the 3-dimensional biomaterials. And we've already shown that if we take these patches and transplant them on the infarcted heart, we can significantly improve heart function. However, there are different challenges and more challenges that we should address. And one of them is how to really create the natural matrix, or something that really mimics the natural matrix. And for that, what we've done, we took pigs' hearts and we decellularized these hearts, meaning we removed all the cells while keeping the matrix, and then looking at the matrix under an electron microscope and seeing that actually we have one big mess of fibers. We have nanofibers, we have microfibers, we have aligned fibers, randomly oriented fibers. And initially we wanted to evaluate what's the role of these fibers, of these fiber populations. So synthetically we created these fibers, the micro, the nano, the randomly oriented, or the aligned fibers. And we seeded cardiac cells to evaluate what's going on, how they assemble into a functioning tissue. And this is one example, and I wish we could turn off this slide here. And you could see, if we would do that, you could see that the cells are aligning on these aligned fibers, and on the randomly oriented fibers, they are just positioned in a big mass. And this is very important because when we're talking about hearts, we want to have a nice anisotropic conduction. We want the cells to start the electrical signal in one point and transfer it to a different point, and not just, something that will beat asynchronously. Another effect that we studied is the effect of fiber diameter. We synthesize these, these nanofibers, micro or even larger fibers. And initially we thought that the cells will prefer to grow on the nanofibers because this is how they grow in the natural microenvironment. However, when we grew the cells, we could see that they haven't developed on the nanofibers, and they really like the bigger fibers. And eventually, when we take everything into account, we can create a cardiac patch that contracts nicely in the lab. This is a few centimeters in diameter, few millimeters in thickness, and it contracts nicely in the lab for a long period of time without external stimulation. However, when we take these cardiac patches and transplant them, since we made the matrices from synthetic materials, they will provoke immune response in the body, and we'll have nice results after a month showing a nice regeneration. And after 2 months, these cardiac patches will be rejected. So the next thing that we've done was to develop another technology that relies on the patient's own materials and the patient's own cells. So we could take fatty tissue from the patient just by a simple 3-minute procedure, take the cells, reprogram them to become cardiac cells, take the material and build the scaffolds from them, and then we have an autologous patch. It looks like this. These are the nanofibers that we get, and the cells really like these fibers. They are elongated and aligned. And we can take these cardiac patches and transplant them. However, they will not provoke immune response. However, they will simply die because there are no blood vessels within these cardiac patches. And this is the next project. It was to create blood vessel networks inside these cardiac patches to be able to bring oxygen into the cardiac cells to maintain their viability. So this is one story. Another story that I will, and we'll come back to this story in a minute. Another story is the transfer of the electrical signal within the cardiac patches. So this is a typical sponge for growing cardiac cells. So the cells are organizing in these pores, as you can see here. And these cells contract and these cells contract, but they're not really speaking with each other on the electrical level. And we thought that if we could synthesize gold nanowires and embed them inside the biomaterial, the cells will start to communicate through these gold nanowires. And I will not go into the details. I'll just show you 2 movies without gold nanowires and with gold nanowires. We stimulated this area and we wanted to see the propagation of the electrical signal. So this is what happens without the gold. There's illumination here and there. This is calcium imaging within the cardiac patch. Nothing is really beating synchronously, but when we stimulate the nanowired cardiac patch, and not here, somewhere over here, really far away, we can see that through these gold nanowires, we're able to get a nice contraction throughout the cardiac patch. So we even optimize this process when we have, this is one fiber with tiny gold nanoparticles, and we could see that between 10 to 40 nanometers we got the best contraction. And remember, we had the autologous cardiac patch, the autologous material from the patient. We thought maybe we can even take this material and decorate it with gold nanoparticles. Here you can see without particles, 4 nanometers or 10 nanometers particles. You can see the islands of the gold. You can see them here. And the most important thing in this slide is this graph showing that we can take a completely biological material from the patient with all the biological motifs within, and create a fully conducting material that will transfer the electrical signal. And when we culture the cardiac cells within, in every parameter, every functional parameter, such as the contraction force or the excitation threshold or the velocity of the signal transfer, we could see that the cardiac patches with the nanoparticles, gold nanoparticles, were superiors. We took it to the rats. We induced heart attacks, and of course we transplanted the cardiac patches on these infarcted hearts, and we could see pretty soon that there is a nice integration on the electrical level between the healthy part of the heart and the cardiac patch. So here you can see that we're transplanting the cardiac patches. You can see the sutures, and we thought maybe we can develop a different technology that doesn't need even to, we don't even need to transplant the patches. Maybe we can inject the patches. And we created this technology that we took the patient's own material and we milled it to small pieces. We found the right recipe to make it liquid in room temperature. Now we can mix it with the cells, and in 37 degrees this becomes a hydrogel. It solidifies, so basically we can take it injected via catheter directly to the heart without the need to open the chest. So these are just some mechanical properties and stability and the nanofibers in this hydrogel. So we took, I told you that we took the material from the patient, and I told you that we took some fatty tissue cells from the patient, and we reprogrammed these cells to become cardiomyocytes. As you can see here in the dish, they really contract nicely. They have all the protein expressions of cardiomyocytes, adult cardiomyocytes, such as troponin or actinin. And eventually we could create a cardiac patch from the patient's own material and the patient's own cells that will not provoke an immune response, and it contracts nicely in the lab for a long period of time. And actually we're currently in the middle of pigs experiments. This is an essential step before going to clinical trials. So we're injecting these materials with the patient's own cells. Now it's in pigs. Another thing that we can do with the hydrogel, this special hydrogel, is to 3D print it. So what we want to do, we have a great printer in the lab. What we want to do is to create blood vessels, as you can see here, within the cardiac patch. So we seed cardiomyocytes in the patch and endothelial cells that will build blood vessels, and then we can take these patches with the blood vessel, lift them up, and even transfer liquid through these cardiac patches. And I remind you, these are patient-specific printed cardiac patches. Other things that we can do with these materials is create nanoparticles to deliver growth factors, or by using microfluidic systems, create droplets, then collect these droplets. These droplets are with the cells. We can collect them, and you can see the cells inside these droplets, and then we can inject them to the organ that we want, either the liver, the brain, the heart, and many other organs. The last project that I will speak about is what we call cyborg tissues, and this is the integration of nanoelectronics with engineered tissues, and not just because it's really cool to integrate electronics with living cells, because there's a necessity. We wanted to be able to evaluate the viability or the contraction of the cardiac cells online. So we created thin films of nanoelectronics with many field effect transistors here. We incorporated them with the three-dimensional biomaterial to create a three-dimensional scaffold with tiny sensors within. And once we cultured cardiac cells, we could immediately see the function on our computer. We could add drugs and see immediately how the drug affects the cardiac cells. But it was not enough for us at this point. It was a great publication, and we really liked this project, but it wasn't enough because, okay, we could see that the cells are contracting or not. We could see if they're synchronized or not. But if they're not, we couldn't do anything about it. So the next thing that we decided to do was to create a new type of electronics that, in addition to the sensing, will be able to provide electrical stimulation to the cultured cells, to the tissue, or to release drugs just by typing on the computer. So if you think about it, the patient is sitting in his house, he's not feeling well, there's something wrong with the patient's heart. Immediately the physician can log onto the computer, see the condition of the heart, and start releasing drugs or provide electrical stimulation. So this is the recording. There's nothing new about this. However, I'll show you now 3 movies. I'll go fast because I see that Ori is becoming impatient. So this is the contraction of the tissue, normal contraction. And now I'll show you what's going on when we decide that we want to interfere, and just type on the computer and we can paste the tissue from afar, 1 hertz or 2 hertz, as you can see here, and this is just quantification. And we can also control the signal propagation. So we have a tissue that contracts from here over here. We can do something like that, just reverse the signal. This is the heat maps of that. And the last slide that I'm going to show is how we can control the release of factors. We can either release positively charged molecules such as proteins. In this case, it was SDF1, a molecule that recruits stem cells into the patch. This is what we could see. Or we could develop a different electroactive protein that will release a negatively charged small molecule such as dexamethasone, an anti-inflammatory drug, and then we could see that we were able to attenuate the function of macrophages, immune cells that actually they approach the patch to reject it. And this is the group, my lab members, and the funding. Thank you, and sorry if I took more time.
Thank you, Tal, for bringing so much hope to 1/3 of us.
Of Americans. 1/3 of Americans.
How about in Israel?
I don't know.
Okay. 2 brief questions, please.
Assumption that the shell
So you're talking about the scar tissue that is formed after the infarction. So the scar tissue becomes fibrotic. It's actually, it's just collagen, and it becomes a really thin layer of collagen. If we connect the tissue, the engineered tissue, to the healthy part of the heart, just make a gap, or just put it without really creating some kind of gap between the healthy part and the engineered tissue, so we're okay. The scar tissue continues to degrade, and will eventually disappear. So there's no problem with that.
Can you use these methods to fuse nervous cells? I mean, it's been for a long time the problem is once a nervous cell is cut.
So this is exactly what we're doing now with spinal cord injuries. We're taking these cells from the patient. We're reprogramming them to become neurons or stem cells, and then differentiating them to become neurons, motor neurons, and we're creating patches for spinal cord injury. So this is something that is done now in the lab. Thank you.
More questions? If not, thank you, Tel, again.